Hi guys, Mrs. H here. Today we're going to be looking at protein synthesis. So by the end of this, hopefully you'll have a better understanding of what the central dogma is. You can explain the relationship between proteins and genes, understand the role of RNA in the process, and describe transcription and translation. So if we look here in this diagram, what we're seeing here is a representation of the central dogma. This is kind of the staple or go-to picture of the central dogma in some form or another. And so what are we really seeing here? We are looking at our nucleus, and we're looking at the cytoplasm, and inside the nucleus, what do we identify? We see DNA, and it's showing a little representation of going through transcription to make mRNA. And then the mRNA leaves the nucleus, goes into the cytoplasm at the site of a ribosome, and is translated into a protein by building amino acids. And so that is the nuts and bolts of the central dogma. And so if you can, at the end of this, look at this diagram and explain the diagram in details, then you probably have a good understanding of this. And so I always like to use this picture to help me go through even, now to describe this process. And so this is critical for all living things because we need those proteins. And where do we have the instructions for building those proteins? It's on the DNA strand. And mRNA is the messenger or the helper that allows the DNA to have this happen. And then the ribosome would be the factory or the site. So we know that DNA is made up of nucleotides. But what's really important about those nucleotides is the sequence of them. What order do those bases appear in? And the order of the bases is the gene. It is direction. It's just like a written language. Like I can put THE together and that becomes the. So if I put A, G, C, A, G, whatever you have, that will become a protein. That becomes the gene. And so each protein has a specific gene or directions or recipe. I like to think of genes as a recipe. It's your, how do I make? How do I make what? Well, how do I make a hemoglobin? How do I make the pigment for my eye color? All of those things are on a gene. All of those things are in a recipe. And so when we're looking at making the protein, we can say that the gene is more precisely or more accurately the sequence of bases on the DNA strand that scribes or directs the cell in how to make the protein. And it's its own language. It's like a spy movie where they have a secret code and you have to decipher the secret code. Well, we do know what the code is. We have ciphered that. We are in that point in our technology that we understand what the DNA, or starting to understand what the DNA language is. We do know what the language is for each amino acid. Maybe we don't know all the nitty-gritties for a recipe. So for example, maybe we know how to make banana bread, but maybe we don't know what grandma's secret recipe is. And so that's kind of where we're at, is we do understand how we scribe for amino acids, but maybe we don't know where all of these genes are located on the gene. And that's kind of what that video we saw yesterday was all about. So when we're looking at protein biosynthesis or protein synthesis, we're looking at two stages. We're looking at the writing or rewriting of the RNA, and then we're looking at decoding the mRNA strand into a protein. that's the second stage. So the first stage right here, this is transcription, the writing, and then the coding, that's the second stage. This is the translation. This is when we're actually reading the secret message. So copying the message, decoding the message. Here's another way that we look at it. This diagram, we see the DNA transcribed into RNA, then it's translated into protein. So DNA is rewritten into RNA, RNA is decoded into a protein. So one of the important things to understand is how do we get this secret code? And it really lies in a codon. And so codon is a sequence of 3 nucleotides or 3 base sequences on the mRNA strand that specifies a specific amino acid. So just like ACT specifies cat, which is this fuzzy little animal that I don't know how I want to describe a cat, but you can, visualize, have an image of what a cat is and know what it is. Well, the same way, ATC signals or specifies a specific amino acid. Now remember, amino acids are the building blocks of your proteins. Remember, nucleotides are your building blocks of your DNA. So those building blocks make these structures. But if we dive into those building blocks of the DNA, they're actually sending us a message. They're sending us directions on how to make the protein. How do we read it? Well, I have to read 3 letters, and those 3 letters will specify the amino acid needed in order to make this. Now, all proteins begin with one start codon, and that's AUG. This is universal. This is in all organisms all through life. So all proteins begin with this start codon, AUG, and it is a specific amino acid, and that's methionine. And methionine is that's the only place we'll ever see methionine is at the start of the building of a protein. So this is the decoder, right? So if you're a spy and you need to get the secret message and break the secret message, you need your decoder. This right here is the codon decoder. So we can look at all of the different possible sequences between A, U, G, C. Now remember, it's U because now we're on the mRNA strand, not the DNA strand. So those 4 bases can be in 64 different combinations. So if I went through all the different combinations of those 4 letters and what they could be in, they're all right here on this chart. And all of them represent an amino acid. Well, if you notice, there's 20 amino acids. So some codons code for the same amino acids. Some don't. It just depends. Do we know why? No, not really. There are stop codons, if you notice. The UAA, UAG, and UGA, this tells the ribosomes to stop making your protein, that you're finished. So just like we have a start codon, we also have a stop codon. So how does this all work? So what are the roles of the ribosomes in this process? So our messenger RNA is basically a messenger. It's just that. They are the secret spy that are going into the DNA and they're receiving the message of, you need to make this protein. You need to scribe for this protein. Remember, DNA has to stay in the nucleus. So we want that in the lockdown, in the vault, so that it doesn't get destroyed. We don't want it damaged. So we keep it in the safe so that it doesn't get ruined, so it doesn't get damaged. So we need some kind of signal or message to send out to our cell. Another reason why is, why make a copy of the entire recipe book if all I want is how to make spaghetti and meatballs? I'm just going to copy that recipe. And so that's what happens. An mRNA is a sequence on the DNA strand that is specific for a gene for a protein. Now, the ribosomal RNA, that is actually a component of the ribosome. Like, it's made up, the ribosome is made up of the ribosomal RNA. So they're important in transcribing. They're going to help read the message. And then the transfer RNA, this is like your truck or transfer. What are they transferring? What are they moving around? They're moving amino acids in the cell. And so they bring the amino acids into the ribosome so that we can start building the protein that we need. So now that we understand the roles and what's going on, let's actually look at each step in some detail. So transcription, remember, this is making the copy of your DNA. So this is literally making the mRNA strand. So I have a protein I need to make. I'm getting a signal that I need hemoglobin, so I need to make an mRNA that will help me make hemoglobin. And so how does this work? So first what happens, excuse me. So the first thing that happens is that the strand of DNA, there's going to be non-coding strands, and the mRNA molecule is actually made by the RNA polymerase, not a DNA polymerase, because DNA polymerase makes DNA. RNA makes RNA, right? So RNA polymerase makes RNA. So the mRNA is known as the primary transcription. So this is what's primarily being made. So if you look in this diagram, that big purple bubble, that's the RNA polymerase. And so it looks similar to the DNA replication, right? So see, we have the big bubbles, we have these proteins that are helping keep it open. Notice we're seeing a DNA RNA. Okay, so instead of DNA being made, we're making DNA RNA. And so what happens, instead of the polymerase adding DNA nucleotides following that base pair rule, we have the RNA polymerase adding nucleotides following the base pair rule. And so where does this begin? What's the beginning process? So just like we saw in DNA replication, we have initiation. So that RNA polymerase must bind to the DNA, and they bind to it at a specific spot. It's called a promoter. I don't know why they call it a promoter. It's just called a promoter. And so this is usually found several nucleotides before the actual gene starts the transcription. And so that's where it attaches. So the RNA polymerase attaches itself onto the promoter site. Once the polymerase is attached to the promoter site, this is when the double helix actually starts to unwind. And again, we have the one side of the DNA strand actually becomes the template. So this becomes the one we're going to read. And then several nucleotides after the promoter is where the transcription region actually occurs. So what happens in elongation? So as the polymerase moves across or down the template, the RNA polymerase is actually moving in that same 5-3 direction that we saw in DNA replication. And so it moves down, but instead of, like I said, adding those DNA nucleotides, it's actually adding RNA nucleotides. And it runs down, making copies just as we did, or not making copies, adding nucleotides just as we saw in DNA replication. The only difference, it's the same process. The only difference is we're only doing a small section because it's a gene, so it's only part of the DNA. The other thing is we're not adding DNA nucleotides, we're adding RNA nucleotides, so that we're literally copying the DNA strand. So then termination. So this is a excuse me, what's called just a termination signal. So it's just basically a stop sign, for lack of way to describe it. So the RNA polymerase reaches a stop sign. So they reach a point in the molecule where the RNA polymerase can no longer code. And so the strand is released. Now, this is called a pre-mRNA strand, and it detaches itself from the DNA, but it's not actually a complete, it's not completed. We still have to do some editing to it. And so what happens in the RNA process, the RNA transcribe, excuse me, got a tickle in my throat. So the RNA that is transcribed is called an mRNA strand, and we call it a pre-mRNA strand because it has to be processed. So the primary transcription that newly synthesized, that's all that's saying is, so the new strand that's not quite finished is called the pre-mRNA strand. We have to modify it. There are parts of it that actually don't code and that we don't need, that we need to get it. So it's like when you're writing an essay and you've just got your first draft done, you have to go back and make sure, you have your commas in the right places, you've capitalized where you need to, that everything is perfect before you turn it into your teacher. Well, that's what happened when we made our pre-mRNA strand. It was just a quick run through. We need to now go back and do some editing. So that's why we call it the pre-mRNA. So how is this processed? So during the modifications, we first need to put basically a cap and a tail on. So on the 5N, we actually add a cap onto it of amino acids. And then at the end, we actually add what's called a poly-A tail. So poly meaning many, A meaning adenine, and then tail because it's at the end. So we add a poly-A tail, many adenines on the end. And we add, again, at the beginning, a cap at the beginning. That's one of the things it does. Now, the other thing it does is there's parts known as introns, and introns are cut out. And I know it sounds different because you think introns stay in. No, introns get out. We don't want introns. Introns are unreadable. They don't have any connection. It's the gobbledygook that's left in there that we don't need, that is not essential to the directions or to the recipe. So if you've ever done a blog for a recipe and there's all that gobbledygook at the beginning of it, and you're reading through it and you're like, okay, this is a great story, but I don't really need it. It's not essential for the recipe. That would be the introns. Okay. The exons are what's left. The exons are what's pasted together, and that's the essential recipe. So we put on this poly-A tail, we put on a cap, so we dress it up, if you would, and we get rid of what we don't need so we look nice and pristine and crisp. And so we're left with these exons in the middle. And so now this strand is ready for processing. And so where does it go? The next step is for that now finished mRNA strand to go and be translated. And so this leaves the nucleus and heads into the cytoplasm and finds the ribosomes. So remember, I had mentioned this before. There are more codons than there are amino acids, right? So there's actually 64 different possible codons of those 4 nitrogen bases. 61 of them are specific to an amino acid. And then remember, we have 1 start and then 3 stop codons. So that's just kind of a recap on those. So what's going to happen here is the translation, we're going to be reading those codons to determine what amino acid needs to be added. And that's what's happening in translation. The ribosome is reading the mRNA strand to determine the codon and they read the codon, and then they go and ask for some help and say, hey, this is the amino acid I need. Come bring this amino acid in. So let's look at, again, the stages here. So we have initiation. So what we have is the mRNA is actually going to create an initiation complex. And this initiation complex is a sequence of the mRNA being locked into the ribosome with the first transfer RNA. So that tRNA and the mRNA are going to actually lock in together, and they're going to lock in together temporarily in the ribosome. And that's the initial complex. This happens every single time at the AUG marker on your mRNA strand. So you're never going to start this initiation complex unless you have an AUG on your mRNA sequence. So what happens after this complex begins? So the next step is the mRNA is read by matching the sequence. So the ribosome actually kind of acts like a place for this to happen. And so, my lights went out. I forgot, I got distracted with my lights went out. So we have that first initiation of the first codon, and actually on the tRNA, it's called an anticodon. So they lock in on the ribosome. So now we have another tRNA that comes and matches up. Well, how do we know what tRNA comes and matches up? Well, we have to read the mRNA, and we have to find the complementary bases that can lock in. So I kind of like to think of it as a key. And so my key opens my door. And so that's the only place that I can go. Well, the anticodon on the tRNA only has one place it can lock in. And if it can't lock in, it's not going to lock in. So for example, on this one, on my mRNA strand, I have UCG. Well, U is complementary to A, and then it follows through AGC. So those can lock in. Now, if you notice on the other side or on the back side of the tRNA, it has an amino acid. And so the amino acids are going to actually form a polypeptide bond between each other. So the two tRNAs lock in, and when they lock in, the amino acids bind together, and now they're bound together. So the first amino acid is released from the tRNA, and it then is released from the mRNA. Now another tRNA comes in, the next one down the line. And so the next one down the line bonds, the two amino acids bond together. It's released from the first tRNA. The tRNA is released, so another one comes in. So this continues on down the line. So then termination. So this continues on down the line until we reach that stop codon. So the stop codon is going to tell us, nope, there's no more amino acids to bond to it because the stop codon doesn't have an amino acid. And so it was called a release factor or a release codon. And so they actually release the amino acid from the tRNA and from the ribosome. And so that complex then kind of breaks apart and breaks down because you have translated all that you need to on that strand. So we're not quite finished because the amino acid at this point is a long chain. And so proteins are not just chains, they're actually three-dimensional structures. And so the protein will then start folding on itself and actually start to construct a three-dimensional structure. So this is the nuts and the bolts of transcription. I hope this helps you understand transcription and translation better, or the central dogma. Remember, if you have any questions, don't hesitate to reach out to me. Bye, guys.